Today it's my pleasure to talk with Professor Yuri Buzsaki. He's a professor of neuroscience and neurology at New York University School of Medicine. He has made just amazing contributions to understanding various aspects of brain function and the interactions between activity in neurons in space and time, and how that is related to and probably very important for cognition and how cognition works. You know, a lot of his work has been in animals, but it turns out the circuitry is pretty much the same, and he can talk about that. Okay, so anyway, the title of this podcast is Brain Rhythms, Neural Syntax, and the Emergence of Cognition from Action. So welcome, Yuri.
Thank you. Good meeting, Mark.
And you grew up in Hungary, and you went both to medical school and then got your PhD there. Can you talk about, you know, when did you really get interested in the brain? Was that one of your first interests in science?
It was about a half an hour decision. I went to medical school. I was completely lost. My previous life was in engineering. I was very much interested in telecommunication. I was a radio ham. And then I particularly was interested in the Morse code, feedback, feed-forward regulations, and things like that. And I had to learn the unending stories about ligaments and muscles and bones in medical school that turned me off. But then Professor Grashtian, who became my mentor later on, gave a fantastic talk about the brain. That was an introduction to brain physiology back then. There was no neuroscience, and I was hooked. So that's it. From then on, I had purpose in my life.
That's interesting. As you know, at a lot of universities, there used to be departments of anatomy. And as you said, there were no neuroscience programs, no real training in neuroscience. And then so what a lot of universities did, including one I was at for a while, University of Kentucky, they switched the name to Department of Anatomy and Neurobiology or something in that. And then, yeah, now most universities have a neuroscience department. It's obviously we're biased, right? But it is. I mean, from an evolutionary standpoint, from a human standpoint, it's the most important organ we have.
You need a certain number of people to form a club. And that club was formed initially in the United States. And the first department of neuroscience was at UCSD. In my alma mater in the university, there is still no neuroscience. It's still called the Department of Physiology.
Yeah. And you were, okay, so your PhD work, so did you start doing electrophysiology then? You learned electrophysiology during your PhD work? Is that true?
It was kind of the MD PhD program in the United States. You go to medical school, and if you're interested in science, you find a mentor and you start working there. So I spent most of my life in the Department of Physiology or in the Brain Research Laboratory, taking exams in other disciplines such as internal medicine and things like that on the side. But I was sure I want to study the brain.
Okay. And then you went to, right, so you did a couple postdocs. You mentioned US.
I went to many places in the world. First, I went to Texas, then I went to Canada, 2 places, Calgary and London, Ontario, back to Hungary, Sweden, then back to Hungary, then UCSD, California, Rutgers University, and then Bayou. This is my zigzagging. I built 19 laboratories in my life.
Oh my God.
And okay, I was, so, this is an important message for younger people.
Yeah.
I never had a tenure track job. I was a floating postdoc, and I got a full professorship from a postdoc.
Wow, that's unheard of nowadays.
Yes, but it's not unheard of. Lee Nadell and John O'Keefe, they were also kind of floating people.
Yeah.
Or Paul Erdős is the most famous mathematician, who never had a job. Or
Yeah.
he
Okay.
didn't want to have a job.
All right. And so your work out, you've done so many things, but you kind of established the synaptic basis of theta and gamma rhythms. As our people will know that from EEG recordings, electroencephalograms. There's these uh, essentially oscillations with different frequencies, and then also these sharp wave ripples. So can you talk a little bit about your interest in these brain rhythms and kind of some of your key findings along the way?
Well, as I said, I started out with interest in radio frequencies and different bands and what they do, and it was amazing that you found them in the brain. Back then, I was scared to call it oscillations because it was too technical. So, in fact, the first person who used the word brain rhythms or brain oscillation rather than rhythms was Mircea Steriade, a good friend of mine, who didn't know much about electronics, but for him it looked like an oscillation, so he called it oscillation. From then on, I also picked up the word, and then it's spread. So theta activity, I inherited the observation from my mentor, André Groschan, who was the first person to observe that theta in the freely moving cat is associated with some kind of behavior. He thought it was the orienting behavior. So that was a good correlate. These oscillations are debated, obviously, because it gives you a glimpse, a 64,000 feet view of the brain. It can provide you an interesting insight what the brain might be doing, but you have to admit it that the LFP, the local field potentials that you record, is not used by the brain. It's a bit like the heart murmur, which is a very good diagnostic tool for the cardiologist, but the heart doesn't use murmur for anything. On the other hand, back then, I used to say that LFPs are an aggregate measures of neural activity. It's like a noise in a stadium that doesn't give you what is the content of the conversation, but it gives you perfect timing. And that is so damn important because timing is what is used in every kind of communication. You have to package information into short, longer messages, and communication becomes communication only when the sender and the receiver has a cipher. They speak the same language. You need a cipher.
Yeah.
If you don't have a cipher, if you don't have an agreement, you don't know how to decipher the message.
Yeah.
Now, many years later, the good thing is that I worked on this oscillation to figure out and to find, with many other people, of course, is that every single oscillation that we know in the brain, at least in the usual frequency bands, are based on inhibition. Now, inhibition is a natural mechanism to give a full stop of a message. So this is when I began to think about, how messages are sent across different structures and how the receivers know when is the end of the sentence. When in human communication, you need to wait until you got to the end because of the sentence, because the last word can change the meaning completely. So it is the inhibition-based oscillations are packaging information in a way that there is a longer, shorter snippets of messages, and then you can concatenate them. So along the way, we learned that it's not only there are different type of oscillations in the brain, but in fact, they form a beautiful hierarchical structure, which means that the phase of the oscillation is modulating the amplitude of the faster one. And the phase of the faster one modulates the amplitude of even faster one, and then it perpetually goes from one level to the other level. Now, this is a beautiful way of packaging information, and for me, the shortest package is a gamma cycle. It's about 10 to 30 milliseconds. And it just happens that it coincides with many other things, such as the time constant of the membrane, spike time independent plasticity, the time constant of the AMPA receptors, the time constant of the GABA A receptors, and so on. So it's a perfect way of sending one message that you can call it a neuronal ladder. And this could be concatenated by the phase amplitude coupling of the theta cycle. So now we can have ladders that last about 7 ± 2 ladders that you can call a word, and so on and so on and so on. So this is what happens in your brain now and in my brain because we are alert. And when the brain relaxes, so to speak, ethologists and also Sherrington, they talked about two brain states or two antagonistic patterns. One is the preparatory state, the other one is the consumatory state. In other words, the brain or the body initiates a movement, and there is an exploration, and that it has to come to an end. And the end is characterized by the different pattern. The brain switches very quickly, and the entire goal of the preparation or preparative movement or exploration is to have a terminal behavior. The terminal behavior could be immobility, sleep, or consummation of food, sexual behavior, and so on. So it turns out that the brain indeed follows these patterns, and then it goes from theta to another pattern. And this other pattern is nothing combined with a big burst, which we call sharp wave ripples. And interesting thing that they are self-generated. They are not initiated or triggered by anything. They are in fact released when the subcortical neuromodulators decrease their grip on the hippocampal circuits. They release them, and all of a sudden, this irregular pattern sharp waves occur. Now, go ahead.
Okay, so the core circuitry doesn't matter what brain region, hippocampus, cerebral cortex, it's excitatory glutamatergic neurons. And then as you're talking about these inhibitory GABAergic neurons that put the brakes on. And then obviously the hippocampus communicates with the cortical circuits. And I think this is one kind of thing you've really provided some insight into is, there's all this work on the hippocampus for a lot of reasons. One is that you cut a slice of the hippocampus and you look at it, you can see actually with your naked eye or under a dissecting microscope, you can see the C-shaped layers of the pyramidal neurons and the dentate granule neurons, and that circuitry is all kind of intact within that slice, right?
Not quite.
Well, but anyway, it's I think it's one reason that the work started there.
Exactly.
And then plus the work showing that lesions of the hippocampus impair, short-term memory and so on. But then the big question is, I'm still trying to get my handle on all these mechanisms, but one of the big questions you're trying to answer is, how is this communication working in the time scales like you mentioned in terms of short, long-term memory, then recall? Is that right?
Yeah.
Yeah.
So what I wanted to say that now we know that sharp waves can occur and the rest of the brain can switch from one side to another when the levels of neuromodulators, then we have measurements for acetylcholine and a little bit on oxytocin, maybe norepinephrine. In my lab, we have acetylcholine. So when the acetylcholine levels are very low, this is when you have a sharp wave. If you activate the medial septum and the cholinergic neurons that you are so familiar with, just a little bit increased, sharp wave ripples are abolished. They don't occur. So this is a special state that is enabled by the fluctuation of these subcortical neurotransmitters, and how the neocortex can affect this self-control and make this change is an interesting thing. But by and large, the very simple and obviously kind of false model is that while we are awake, we are learning, and this is when your level of consciousness is high, the working memory is working perfectly, but then you have to remember certain parts of this. So the interesting thing is that unless you have to see this interaction with you many times, what can you remember from our conversations tomorrow? And this is what we are very interested in, and we can tell you about it. What is the selection mechanism that allows you that certain things will be entered to storage and other things will be put in the trash can? Now, it turns out that the key thing here is the sharp wave ripple. And the old model suggested that while we are awake, we are learning, exploring. And when the terminal behavior, that is the reward, you can call it the backpropagation algorithm or something like that, that at the end there is reinforcement. Reinforcement learning is a big thing today. Then the sharp waves occur. And then the synaptic changes that occurred during learning will alter the circuitry a little bit. And this altered change will be then broadcasted to the neocortex while you are in the consumatory state, including sleep. And it was an interesting thing that, humans in the 8-hour sleep have about 4,000 to 6,000 of these sharp wave ripples. And back then it was an interesting pattern. This is the most synchronous pattern in the mammalian brain. So that alone was attractive to me. And it seemed that it's good for something. Yeah. Now, but you just mentioned the slice preparation, LTP, and those things that are forgotten these days. That people were searching for mechanisms in the in vivo brain, how this LTP inducing trains, which back then was, you may remember, 100 to 200 hertz, tectonic pulses or short tectonic pulses could bring about something. So this was the era in which I was in, and all of a sudden I see these sharp wave ripples. The sharp wave is a reflection of the output from the CA3 region. The largest sharp waves can occur or can be recorded from the middle of the stratum radiatum, which is where the sharp wave collaterals converge to. So that's a sharp wave. And then these excitatory inputs from CA3 excite both the pyramidal cells and the interneurons, and their negotiation at this very strong depolarization level gives you a fast oscillation, which is about 100 and 200 hertz, the ripple. So it's a complex sharp wave and ripple. This is why I call it sharp wave ripple. Yeah. Now, there was this 100 to 200 millisecond pattern, which was exactly the same kind of pattern that people use in the slice preparation. I said, oh, if you are looking for an LTP mechanism in the brain, there we go. So then we added the speculations and so on, and we observed very interestingly that when we do an artificial LTP in vivo, in a funny situation where we cut the familiar phonics and so on, then you record from a large number of neurons, but large back then was 16 sites, and then you got a signature of the pattern. And that signature of the pattern of the stimulation, interestingly, was expressed later on, spontaneously.
Replay.
The sharp waves change their forms following the pattern of my stimulation. And then I changed the stimulation polarity and stimulated somewhere else in the internal cortex, and then I got another one. And so obviously it was a trivial thing to assume that, oh, you can change this pattern and it spontaneously replays. And back then I knew that the replay occurs. We didn't call it replay then because we didn't know what is going to be replayed. I just looked at this artificial replay. And then later on, Matt Wilson and Bruce McNaughton showed it before us, that indeed such a thing can happen. And then there is recapitulation of the learned information, at least when it's read out by pairwise cross-correlative methods. So that was the initial idea. And many years passed by and lots of speculations have been on sharp waves, but there are many problems. Then we can talk about the problems.
Okay, I had one question about the subcortical inputs and the acetylcholine, cholinergic and noradrenergic and So you were talking about how the memories are formed when we're awake, right? And then, so we know that you tend to remember things that have some emotional context, right? And some, certainly the noradrenergic inputs from the locus ceruleus, they've been kind of connected with, if you, I think if you enhance those, you can like enhance short-term memory or something like that. So then during sleep, what's the status of these subcortical inputs in terms of their activity, and is that consistent with the idea that during sleep there's this memory consolidation going on?
Absolutely. So the interesting thing is that all neuromodulators that we are aware of, including social neuromodulators, so to speak, oxytocin, which we are routinely measure now, they have an interesting fluctuation. So the big revelation of the past 5 years, when we started to the tools to monitor them in a fast manner, a sub-second scale, which was needed to make the conclusion what I concluded, that at the minimum of the sharp variable maximum happens at the minimum of acetylcholine levels. You had to correlate this. Now, during sleep, all the subcortical neuromodulators go down. And in fact, they go down in a beautiful manner. The non-REM sleep is not a flat thing. It fluctuates every minute in a rat. It's called the microarousals. And so every minute you go from a shallow arousal pattern, if you want, to a deeper one. And then you can see how the acetylcholine level decreases, how the excitability of the system increases at the same time. And then at the very end, the sharp variables start popping up when the acetylcholine level is low. And then the sharp wave, the acetylcholine goes up during the microarousal, and then you start the whole process again. Now, the other neuromodulators, all the other neuromodulators are behaving or working together with acetylcholine until now. But then when the non-REM ends, it has two choices, or three choices actually. One is this microarousal that is super important for subjective feeling of sleepiness, not sleepiness, that you have a subjective feeling of good sleep. If you are perturbing these microarousals, then you wake up and said, "Oh, I didn't sleep at all," even though you were in bed for 8 hours and you lost consciousness. But now, if you look at the neurotransmitters, then you can come to this microarousal, REM, or waking. Now, when you switch from non-REM to REM, then acetylcholine goes up pretty high. All the other neuromodulators, all of them, go down. So that's
when you're saying that transmitter, are you actually measuring the level of the transmitters or the synaptic activation of a cholinergic synapse?
So today you can do both. You can do both. This is the GRAP technology that is spreading. Yulong Li is a big factory for this, and now there are probes for almost everything. And there are other methods also, not only Yulong Li.
And I missed it. So these can be measured on like
this is fiber photometry.
So it's over tens of milliseconds. That
exactly tens of, I would say, sub-second.
Okay. Wow. All right.
Yeah. So this is fantastic, right? Because now you can really look at what is the time relationship between a interesting pattern such as sharp wave and the rest of it. I use sharp wave as my anchor because you can do 5 different things. We can measure 2 neuromodulators simultaneously now, a little bit with more complications, perhaps more. But if you have a time 0, then you can measure any neuromodulators in different animals. And then you can look at the temporal relationship of the neuromodulators to each other using the sharp wave ripple as your anchor. So this is about how much we can say about the relationship, but this is a fascinating think that there are these allowing patterns that sometimes they work together, sometimes they work in an antagonistic manner. In your days, or which is my days, you may have wondered, like I did, why do you need two neuromodulators? Why, or more? Why don't you just have one? And then it does the switching for you between preparative and consumatory behaviors. Now it seems that it's a nicely orchestrated pattern, and it follows the needs, and exactly what the needs are of the circuits we don't know, but there is plasticity, transmission, and lots of things, and different neuromodulators have differentially innovate different parts of the neocortex, hippocampus, basal ganglia, and so on.
Yeah, that's really, and so I imagine serotonin's also playing a role.
Absolutely.
Yeah.
Serotonin has to go down. REM sleep doesn't have serotonin. So that's a fascinating thing about REM sleep. There is no modulator, but it looks like a waking pattern. So REM sleep is an interesting thing from the memory point of view. I'd like to go back a little bit in the history. So the idea that we replay our learned information during sleep is probably coming from the Greeks. I don't know, but the real formulation of this was, of course, Sigmund Freud. Sigmund Freud thought, oh my God, you are dreaming, and therefore we can interpret your dreams and so on. And so it was persuasive in the layman society as well as among neuroscientists that dreams are important. And then when REM sleep was discovered, then the relationship between dreaming and REM sleep was established. Then all memory researchers zoomed into non-REM, into REM. Sorry, that was the big thing for many years. And they did all sorts of interesting experiments that can be redone today with better methods. But then I discovered the relationship between sharp wave ripples and LTP. Said, my God, this is perhaps more important plasticity, and non-REM is about 80% and REM is about 20%.
Yeah.
Maybe that's the key. So our work shifted the emphasis for memory consolidation from dreams to actually when there are no dreams, or very little dreams. This is the non-REM pattern.
Now I'm thinking about people that have insomnia, having trouble sleep, and I think you're interested in it's kind of a new age of using the drugs. I've worked a lot on Alzheimer's disease, Parkinson's disease. There's no drugs that really affect the decline much at all, and as you know, a lot of big drug companies have, and it's also true with psychiatric disorders, a lot of big drug companies have completely quit pursuing drugs because they spend so much money, their view is we wasted it. But now I had a podcast with, gosh, I'm terrible on names, Yuri.
For a good reason, because names are human inventions, and it's a completely arbitrary connections between natural memory and arbitrary things such as names. I could be Mark and you could be Yuri.
Yeah. Okay. Anyway, I had a conversation with an investigator that does a lot of work on brain stimulation for depression, and transcranial electric or direct current stimulation. And so you think in the future, people with insomnia will be treated by stimulating in ways that mimic or induce the right patterns and amount of deep sleep and REM sleep.
So you're probably talking about the work of Helen Mayberg about depression and deep brain stimulation.
Yeah.
And of course we are very much interested in that work. Yes, the problem with drugs usually for sleep is that sleep is a symphony. Sleep is a beautifully orchestrated symphony. It's not just you turn on sleep and then you're sleeping and then you wake up. It's not a binary process. It has a lot of patterns. It starts out drowsiness, then you go to different levels of sleep. In the old days, we talked about stage 4 sleep. We now talk about slow wave sleep, which people, not you and me, but regular people get around 2 o'clock in the morning. So it takes about 4 hours to get to this deepest stage, which lasts for about half an hour, which is super important because this is when there are tons of sharp waves and a lot of growth hormones. In fact, 95% of the growth hormones are released in this half an hour. And growth hormones are important for protein synthesis, and protein synthesis is important for long-term memory. This was another reason why I love sharp waves, is because you need a lot of sharp waves in order to release it. Now, after 40 years of age...
So you said growth hormones. Do you mean growth hormone itself, or neurotrophic factors like BDNF or IGF-1 or those kinds of things?
So again, we don't know exactly because all these measurements were done at the half an hour scale maximum or hour scale rather than a minute scale. But it seems that growth hormones are most obvious. Others, they are demonstrated that it happens in this very short time window. BDNF and others probably have a longer distribution.
So are there structural? Okay, right. So kind of old thinking was, okay, LTP, you mentioned that, if the synapse is activated a lot, that it gets stronger and actually changes size, make it even get bigger. Or over time, there be maybe increased number of synapses. So do you think during this critical period of sleep that if you block that in an animal, if you prevent them from going into that, are there structural correlates in terms of numbers of synapses or something like that?
That's exactly right. But we know from the LTP literature that there are different phases, and the late phase is protein synthesis dependent, because this is where you have to do something with the synapses to grow.
Yeah.
But without growth hormones, there is not much growth. There are beautiful experiments done in sheep. They measure the growth of bones that we grow literally in size during sleep.
Oh yeah, well, we have a granddaughter now, and we've been talking about that. She's only growing during sleep.
Okay, so now the sad news. The sad news is that this critical slow wave sleep starts to disappear at around 40 years of age. Okay? And now even sadder news that you and I don't have.
That explains a lot, doesn't it?
You don't have anything, okay? You don't have anything.
Everything goes downhill.
It's downhill. So we don't have protein synthesis. That's bad, right? But how does it affect our memories also? That's bad, and so on. So there are lots of things that go together. Now let's go back to the disease, such as Alzheimer's.
Yuri, now this is interesting. As you know, I think this is true not just in the sciences, but a lot of disciplines, that people's creativity is best when they're, say, in their 20s and 30s. In other words, a lot, it's not 100%, but for the most part, Nobel Prize winners in science, often the work that they're actually got them the Nobel Prize was when they're in their 20s and 30s. And you get to a certain point, we have a lot of knowledge we retain and can pass it down to our graduate students and postdocs. But what do you think about, do you think you're as creative thinking now? I don't, I'm not. I know I'm not. But I'm wondering what you think.
It doesn't matter what I think. You summarize this very nicely. Very nicely.
That's true. That's true.
I'm studying mice and rats. I just make generalizations that are not unreasonable, that I know that if supposedly, there is such a thing as memory mechanism and what we know about it, then there has to be a system level control of all of this. And it seems to be a beautiful system level because, for the past 30 years, ever since we talked about this two-stage model of memory, we were obsessed, and everybody else is, that how the information is read out by the neocortex of this sharp wave ripple.
Yeah.
Now, other people looked at patterns in the hippocampus in funny animals such as the lizard or birds. And lizard doesn't really have a lot of cortex. In fact, it's a rudimentary, so there is not much reader. But the sharp wave ripples are just like in humans. So to make long story short, last year we had a nice paper in Nature where we showed that these sharp wave ripples do a service to the body. They decrease glucose levels.
Yeah, I saw that.
So all of a sudden you connect it back to a Asian pattern that occurred for one purpose, and only later evolution is smart enough to exploit it or accept that pattern for something else, because it's so nicely synchronous, it can have such a big impact on the downstream readers, which are now huge. And this is another important factor for me in terms of the information processing things or message sending, is because the richness of the message is not in the sender, it's in the reader, in the receiver. The same messages can be decompressed in different ways, and the hippocampus in a rodent can already generate enormous number of patterns, but this value of the patterns is only in the reader. And this is, of course, why the neocortex is so much bigger, relatively speaking, than the hippocampus. And this is the evolutionary rule, with the exception of dolphins and cetaceans.
Yeah, and so the brain has to, not only like encode patterns, but be able to process them and kind of compare them to other stored patterns, like sequences of because we think, as you say, we think in sequence. Everything is important in a sequence. First this, then this, then this, and so, but for this, the sharp wave ripples influencing energy metabolism, I'm particularly interested in that. We did all this work over beginning in the 90s. I finally got around to writing a book.
I know that.
Oh, you know that. Okay. But anyway, so now I'm thinking, I encourage you to do an experiment. I mean, it's obvious, right? You do put the rats on like every other day food deprivation and look at the, use your multi-electrode things. And
well, there are papers actually, malnutrition and sharp waves. Sharp waves are so wonderful because it's a built-in sensor for excitability. No wonder in every single disease model, including your favorite Alzheimer's disease, depression, schizophrenia models, if you believe in schizophrenia model of the mouse, and epilepsy, and they are all always changed. So using sharp waves as a target to restore patterns is a good target, except as you mentioned, Alzheimer's disease research is basically has been molecular biology and pharmacology. Nobody, because I think the reason for that is that in every single disease in the history, you would like to have a shortcut. And the shortcut is, let's try this drug, let's try this drug, let's look at the anatomy and the pathology and see how we can make up some great ideas. And sometimes it works, most of the time it doesn't, because we don't understand the mechanism. But how the mechanisms change during the course of Alzheimer's disease, nobody was interested in, with the exception of a few people in San Francisco. But that's a gold mine because it's slow. You can record activity of various parts of the brain, starting with the different layers of the entorhinal cortex, and seeing how these patterns change over time. And you've got 9 months.
And also that would be some interesting experiment. There are some, they're rare, but there are familial cases.
Going back to nutrition, there are a couple of papers, not in high-ranking journals, exactly for this, because nobody's interested in the physiology, showing how malnutrition affects sharp wave ripples, for example, and theta oscillations and physiological patterns. And then, of course, you end up in a mediocre journal with that, because they said, oh, it is so far-fetched, it's so far away. because there is no intermediate links yet. Same thing applies when you look at things like, obesity, depression, and then the altered biochemical and endocrinological changes or immunological changes. And you said, oh, of course, if you are very, very fat, you can't sleep well. If you are depressed, you don't sleep well. However, what you don't realize is that the things can and likely go the other way around. The first thing that happens is the brain changes, and the sleep is a perfect readout of the changed brain pattern. And the consequence of this is what happens during waking rather than the other way around.
Yeah.
And this glucose modulation of sharp wave ripple was a good example for that we provided a mechanism that, and we identified how you go from the sharp wave through the lateral septum to the hypothalamus and so on. And that is an interesting thing that probably happens also during depression. So depression, all the depression drugs have a tremendous effect on sharp wave ripples also, and SSRIs and tricyclic antidepressants wipe out REM sleep. So we started out talking about this beautiful pattern that goes on, and the drugs that are available, they make you to fall asleep or do something else, but they don't maintain the sequential activity that is called sleep. And sequences are important for the brain at the short time scale and the very long time scale and everywhere else.
So you mentioned okay. So the idea would be to follow longitudinally in humans. What I was thinking of as experiment, there are inherited forms of Alzheimer's disease where the age of onset is very stereotyped.
Yeah.
And so you wouldn't need many individuals to do it. Do a study where you record, I don't know, once a year, beginning when they're such and such an age, and then see if there's alteration in the sharp wave ripples that precedes any measurable cognitive thing. And then is there a way to either induce or block sharp wave ripples in humans? Or
super easy.
Oh, okay.
Super easy. Any drug that increases acetylcholine levels, you can give physostigmine. In fact, Jamborn in Germany have done these experiments, and giving physostigmine like this is cholinmimetics before you go to sleep. So this is an amazing thing, right? That we know that we thought that in order to be smart, we need the cholinergic activation. However, it's good only at one stage. So he showed that when the cholinergic drugs are given before you go to sleep, next day there's a memory deficit. And he speculated, he didn't record from sharp wave, but he knows our work very well, and he says it's probably the sharp wave ripples are suppressed, and he's right.
What about
I know from my own experience when I was in medical school.
What about an antagonist, though? Will that enhance?
Well, you have to be very careful, because atropine, for example, would be, or scopolamine would be that, but it blocks a lot of other things as well. But we are very, very good. We neuroscientists are very good of making the brain work worse. We are not very good of improving it. So when I was in medical school and I prepared for my examination, back then I didn't know much about amphetamine. Nobody knew. I didn't know that it's a dangerous drug. So I just I got from my friends. I stayed up the entire night, and my own experience told me that, my God, something is wrong with my memory, because next day I don't remember anything, even though I kind of had the gestalt that, oh, it is probably on that page somewhere, and I knew it has to be somewhere in my brain, but I didn't remember it, because in order to remember, you have to switch from one state to another. If you don't have that state change, what comes in goes out, rather than getting stored.
This comes to something I kind of experienced myself when I was an undergraduate in college, and it's called cramming for an exam, right? You put off studying more than you should, and then
You don't sleep.
Then you're staying up late at night, and you're thinking the more time I spend on it, the more.
Absolutely wrong.
And it's counterproductive.
Exactly.
Yeah.
Exactly. Sleep is a very valuable thing that we have. Yeah. And I think the society now recognizes it, how important it is, and how much we have to preserve that. And it's, sleep is good for all, memory. Memory is a precious thing, but our immune system is totally screwed up. Yeah. So, but your waking next day is affected. So the funny thing, not the funny, sad thing in the United States is, that there is a drinking age, and then if you, and there's of course, everywhere in the world, if you're drinking, you're not supposed to drive. But, one glass of wine equals with about 3-4 hours sleep deprivation. So, if you are sleep deprived and you party, 4-6 hours and you're driving home with 1 glass of wine, that's about 4 glasses of wine or 3 glasses of wine. And nobody cares. And there is no regulation that, partying young people, they didn't drink, but they stayed up the whole night, because your attention span, your reaction time, and everything is altered. You know it from yourself.
It's probably true with cannabis too, marijuana, right?
Absolutely. Yeah. No, we had a study on cannabis, and then I decided I'd never take cannabis in rats, because it in a nutshell, this is what it did. It changes the timing at the gamma cycle level. And this is where the neuronal letters have to be concatenated into theta cycles. Now, what we observed is that the place fields that are necessary for spatial navigation, the cognitive map or the place map in the hippocampus, was not altered at all. It was the same, but the animals couldn't solve a spatial task after the same dose that corresponds to the recreational dose in humans of marijuana. So, I don't have any objections. Alcohol does the same bad thing, yet it's allowed. All I know is that I don't want to take any cannabis into my brain.
It'll be interesting. Is anybody looking at psychedelics?
Oh yeah, I mean, that's a big thing now.
Yeah.
For a good reason. My wife is a psychiatrist, and she's very enthusiastic about any new drug.
Yeah.
For a good reason, is because none of the drugs, for example, in depression, work for more than 20% of the people, which is super important, because the 20% of the people, it makes a big difference.
Yeah.
But you have to find that one for each patient. Psychiatrists are not working with groups of mice and rats. They are working with individual animals, and then it doesn't work. It doesn't work. And they run out of options. So anything that and it affects only 5% of the population, would be useful. So most practicing psychiatrists are welcoming this. Now, is there any hope for that? Who knows? There are many researchers, including NYU, that are devoting quite a bit of effort into exploring these old drugs.
Yeah, Hopkins is too. They started a whole kind of a small department or program on that. Talk a little bit about evolutionary perspective. So you mentioned food seeking, right? So nowadays our occupations are very different than they used to be during our evolution, right? So kind of the most important thing for evolution is, can you get food and survive? And can you pass your genes on? Can you reproduce? So what's the, and you mentioned the hippocampus and spatial navigation. So obviously, to get food, you have to move, right? Most animals in the wild don't wake up and open the refrigerator door. So how do you see these mechanisms in terms of brain evolution, and how the way humans have looked at this? So for example, and you mentioned this many times in your writing, that when people do functional MRI, for example, in humans, the person is laying flat, completely still. And I know by myself, I do a lot of good thinking when I'm walking, or I used to run. And
Look what you are doing with your
I'd
fingers.
come up with some of my best ideas.
Watch your fingers. Watch your fingers and your hand. While you are talking.
Oh yeah, that's true. I thought you're saying maybe typing is enough.
No, you can't give a good talk when you are paralyzed.
Right. So this is the idea from cognition from action. Can you talk about that? And I guess I should ask you, how much time do you have, Yuri? Do you have another?
It's up to you.
Okay.
So my interest of is evolution. I'm not an evolutionary biologist. I wasn't prepared for this, but I realized that what we are doing is a funny thing in neuroscience, because historically, interest in the brain didn't start out with the brain. We curious people started out figuring out the psyche, and the mind, and then that was the interesting thing, and then the idea that the psyche and the mind was given to us. The brain, we didn't know about. Aristotle didn't believe in the brain at all, and many people, but that this soul that is somehow put into the body temporarily is given to you for one good purpose, is to learn about the beauty of the world and learn about good and bad and learn about God and worship God, which is what good people have to decide, that they side with God rather than against.
And by the way, the animals, lower animals don't have souls. No.
Tell me which is a low animal. There's no low animal.
No, we're just another animal. But that's my point. I mean, back then people
that's
still do today think we're like.
That's exactly right. So this is the historical perspective, and then what happened is that the idea was that the brain is gathering this information like a sponge, and then something happens with that information, because we have to make a decision according to Christian religions, and then only after you made the decision you make an output. So this is the perception action idea. But of course, between perception and action, there is this unknown territory of what you call decision making, and which is a very popular idea today. In the old days, it was called the free will, but it's the same thing. And then you come up with an interesting discussion about what we have to associate. So in the West, in the East, things happened slightly differently. In the West, West means Europe for us, unfortunately, only because we ignore all the cultures that contributed to humankind's knowledge. But then this became British empiricism, and then the British empiricism translated into various things such as associationism and so on. Then, Ivan Petrovich Pavlov, when I was in Hungary, he was the other big thing. He said, oh, you have to associate the CS and the US. It's all passive. The dogs were in a harness, just like today's animals, they are head fixed. Or you mentioned fMRI, were there. It was a fantastic ideology for my system where I was growing up, because it is imposing the government's wish through associations on people who don't have to act at all. But organisms act. In fact, I just want to short circuit the discussion about this, but what I want to say is that we created a lot of interesting words, such as memory, emotions, decision making, and so on. And we discussed back and forth, how they relate to each other. And many of the debates today are pretty much the same debates as they were 2,000 years ago. Because the only way how you can be wrong and I can be right, if there is an arbiter. And for me, the only arbiter is the brain. Now we have to look into the brain. So when people started looking to the brain, they inherited this extraordinarily big vocabulary, and they made up their minds. Said, "Oh, I'm going to work on decision making," and we put ourselves into these boxes. And then, I'm interested in time. I'm interested in space. Then I focus into one structure of the brain. This is the home of those human concocted ideas. and we pretended that those things actually exist, including space and time. But, you know, I'm working in rodents, and then I have a hard time, not me, I mean us generally, to sell that what we observe here is actually relevant to humans. And then this is how we say, is not episodic memory, is episodic memory like, and short-term memory like. And the reason for that is that all the definitions in cognitive neuroscience starts out with the human brain. So, for example, in my field, memory, especially the interesting memory, the one that I can communicate, is called declarable memory. Larry Squire came up with this nice word, it's perfectly right for humans, but it means that declarable memory, which, you know, is in two classes, episodic memory and semantic memory, are the ones that I can verbally communicate, I can declare. And if I can't, then it's not. But rats do not convey any words to me. So
Right.
all of a sudden, the definition that are always based on language, all cognitive science, or almost all cognitive science vocabulary definitions, are based on human language. And that is when I thought, that's not quite okay.
Yeah.
It makes things hard, and we just have to fight forever. They said, oh, I see something in the animal that may relate to that thing. In fact, it should be the other way around, that there is a mechanism that evolution worked out, and its primitiveness looks primitiveness when you look at the very end. But many of these patterns were not made to make language, to make cognition the way how it works in the human brain. So looking at its simplicity informs you oftentimes more than, even if you allow me, and I don't want to do that, you know, put in millions of electrodes in your brain, and you know, if I've had if forget about ethics and everything, we just say, okay, let's put thousands of electrodes or fMRI and do millions and millions of experiments in humans and neglect all the animal experimentations, then it's just a waste of time. Not waste of time, but it's much less effective than using simpler organisms.
Yeah, I agree. I had one conversation with a well-known person that studies emotions, and
exactly.
And that person, you know, I asked him about, you know, animals, and say dogs, or, you know, when I was young, we'd always have two dogs. When one dog died, I mean, it's just obvious to you that the other dog that's still alive is sad. They're exhibiting features of sadness. But this particular scientist said, oh, you shouldn't attribute.
This is Joe Ledoux.
It is. You guessed it.
Oh, he's my colleague.
Okay.
My respected and loved, very good friend.
Yeah.
And he has a point.
So anyway, but he didn't want to go there. But my thinking is like yours. I think it would be incumbent on, say, Joe, to establish that animals don't experience these things, because this seems something very important.
No, just to say, no, Joe is a smart person.
Okay. I know.
He doesn't say that. It's your translation. He says he don't have a good way of objectively saying. He doesn't say that dogs don't have emotions. Of course not.
Okay.
He just say emotions and pain and those things that we call fear. I mean, his
joy,
his
and
list
pleasure.
of fear, conscious experiences. And once you have conscious, the level you are talking about consciousness, that it has to be declared by language. That's another problem. And he says, I don't know, because the dog doesn't tell me. And as long as it doesn't tell me, then it's not there. But my argument is, again, my wife's argument with a psychiatrist: you don't have to talk. Psychiatrists have to listen and watch. And so, you know, when you kick your dog and it starts screaming and saying that it's not really feeling bad, it's it's ignorance.
Yeah.
Right? So yes, he didn't tell me except made a sound and things like that. That's not communication, but you can infer language or language-like communication or non-language-like communication fairly well.
Yeah.
Fairly well. So indeed, emotions can be studied. If not even emotions, you just set up the conditions. How Joe's extraordinary contribution to this field is very large, even though
I know
he tries to dismiss it in an interesting way. I mean, he identified the circuitry and so on. That's super useful.
Yeah. Yeah.
So you don't have to sell yourself as an emotional researcher. You don't have to say I'm studying memory and things like that using these words, because now I'm embarrassed about memory, because now the same substrate that is responsible for memory is basically post-diction. But I can also use the term differently, as prediction. And in fact, the embarrassing thing that these structures that are responsible for memory are also structures for imagination and planning. So these are historical separations of things from each other and not brain-based.
Yeah.
And
so, Yuri, to finish up here, so on my other screen here, I have some recordings. So you have rat that was trained, I guess, to go left or right in a certain maze, and then you put them on a running wheel after they've learned that, and so essentially what you're showing are these internally generated sequences that were, I guess, replayed
yes,
when the animal is not.
it's not necessarily replayed. It's just a sequence there that is needed. In this case, was working memory.
Okay. And there's a right.
So there are two interesting things going on in evolution.
Yeah.
One is internalization of things. So internalization of things means that you don't act out. So, for example, one of the first very clear pattern that we observed was the head direction system. The head direction system is a fantastic thing. Jim Rank discovered this, that you are recording a neuron, and then you turn your head, and then it fires like nuts in one direction, and then it's silent. And then you go to another room, and then it anchors, the organism anchors itself to one corner. That will be your arbitrary north, and then the neurons fire one after the other. So it's a ring of one dimension, and the 60 degrees goes after 40 degrees representation, if you want, and then 90 degrees comes after 60 degrees. So that's a ring in the waking state. Now, what is driving this? Well, I'm looking at the corner, but when I close my eyes, nothing happens. And I fix my head, so the vestibular system is not working. So this has been kind of known subconsciously by my researchers, but they are still thinking about drivers. Now, we wondered what happens to this ring attractor, so to speak, when the animal falls asleep. And what we found is that during REM sleep, we can tell which direction the animal is looking at in its mind or in its brain, because the neurons fire exactly in the same sequence. Now you go to REM sleep and the same thing happens, but the movement of the ring is 10 times faster. It just goes around like this. So it shows that it's an internalized pattern.
Yeah.
And then we generalize this, and we have written a couple of reviews about this, that navigation is what the hippocampus does, for sure, as one job. This is probably, I don't know to say early job, but because in evolution, everything goes hand in hand. But let's say it was worked out with the help of the external environment. And then you disengage from the external environment and you start traveling mentally without the anchors, without the helpers. Yeah. And this is called memory. If you travel back into the past. But if you go forward, that's planning and imagination. So these are the two examples for me for disengaging from the world. Externalization of the brain function is even more important, and it's more human, that you put your thoughts out into the world. You make a tool. And if you walk in a mountain or anywhere, or in the Sahara Desert, you will find an object. You can tell for very high certainty that it was a man-made object. Even if it's a rock that was polished or something like that, because it's a message that was sent to you by a guy, a woman, 40,000 years ago. And the good things about the externalized objects that they tell stories even after you die. You know, when you write your book, which is on your shelf,
Yeah.
you know, it will be there forever, hopefully, and people can learn from it, and you can tell it to many people. So going back to the internalization part of the story, so we ask is that, oh, is it really true that the spatial place neurons, as John O'Keefe calls them, fire because the animal in the XY coordinate of the Cartesian space? What happens when you freeze the animal in space? This is why we train the animal to run in a wheel.
Yeah.
And then to our surprise, it didn't happen what you expect, because what you expected, according to O'Keefe's theory, is that a neuron that fires here and that should fire forever as long as you are here.
Yeah.
And all the other neurons should shut up, right? So what we found is that there was no such neuron. In fact, every neuron had a lifetime, which was pretty much the same lifetime when the animal navigates in the environment. So the conclusion is that these neurons, the hippocampus is not a honorary V1. It is not reflecting inputs. It's making a pattern.
Yeah.
And from this story, we went through various thinking, and then we made the inside out kind of idea, is that the hippocampus and probably the rest of the brain is not learning everything from scratch, but it already generates pre-configured patterns that are enormously large. And learning is not a synthetic pattern, but in fact is a matching between a pre-existing pattern and the experience. So it's an upside down thinking, if you want. I call it inside out thinking, because the brain's primary goal is to maintain its own dynamic. The second thing is to generate an output. And when you generate an output, the brain wants to register the consequences of its actions. So this is how agency emerges with time. And the agency can emerge in time because every single time the brain generates an output, the output can be a variety of different things, such as moving your muscles, releasing growth hormones, but also the thought is an output. The thought is an action. It's a delayed action. The only utility of thought is that it's utilized somewhere later in your life. So it's not by miracle that all the thoughts that people think is in your prefrontal cortex, which is next to the motor cortex. The front part is the action part. And so this is how we kind of made upside down that once you generate an action, that's important because then you can survive. If you are in the right niche, that's another thing that we forgot about, the importance of the environments, that animals are very efficient in their own environment. You put a honeybee into a different situation, it's completely lost. But we humans think to be very versatile and survive everywhere else, so we forgot about that. So once you generate an output, you can survive in C. But then, if you want to perfect it, then you would like to have sensors added to your brain in order to monitor the consequences of your actions more precisely. So the primacy of motor action is the importance. So we are talking now about the action perception cycle rather than the perception action cycle.
And so now I'm thinking of my granddaughter again. She's 8 months old, right? And so obviously she's not talking yet. She can make some sounds and kind of laugh and giggle. And she's not quite walking yet. So she doesn't have language. She's not really moving, but she explores things, right? So, I mean, this is a huge question, right? What's happening in terms of at what point in brain development are these patterns already there when they're born? And how do they change as they start to be able to move through their environment, and then when language comes on board?
So I can tell you what I know from experiments. So we asked the question differently. We asked the question is, what is the first organized pattern in the brain? Okay. And we looked at the hippocampus and looked at the neocortex and looked at the thalamus. And then you can look at it in a newborn animal, I mean, newborn rat, because the newborn rat is approximately has the same age as the human fetus between the second and third trimester of pregnancy. Okay, so now we have a newborn rat. We didn't go earlier. We said, what is the first organized pattern that happens in postnatal day 1, 2, 3, 4? And it turned out to be a spindle. Okay, but the spindle, every one of them was initiated by a kick or a movement in the body. Now, this was an enormous revelation for us, because everybody has known for a long time that baby kicks in the womb are super important. They correlate with your Apgar score and your IQ many years down.
Oh, really? Huh.
That has been known. What we didn't know what service these kicks do to the brain. But then now we know that there are at least 3,000-4,000 movement patterns. People watching with the fetus documents this very well, that sometimes your gastrocnemius twitches a little bit. You can have a hand jerk, or you have a whole body startle reaction. Now, this is exactly, if you ever observed rat pups or mouse pups, they are together. They are like popcorn. They always move.
Yeah.
And so every single time there is a movement from out of nothing, there is a brain activity, and this is a spindle. Now, let's suppose that we have, don't suppose that we know that we have about 620 plus minus muscles in mammals, and you can put the muscles on the table, and those are twitching stochastically because acetylcholine is released stochastically early on. And there are also muscle spindles that go into the spinal cord, and the first organized pattern that occurs are the vertical patterns. The spinal thalamic tract is intact. The thalamocortical vertical patterns are intact. No subcortical modulators are coming into the cortex or hippocampus yet. No horizontal connections are present. Everything is vertically organized. Now, when these neurons switch, the muscles twitch, then it generates something locally, in a localized manner. So if the muscles were on the table, not interacting with each other, that would be a useless teacher, because it would just generate random patterns in your brain, like in a tissue culture.
Yeah.
However, the muscles are anchored to the skeletal system. Now, the degree of freedom of these muscles changes and decreases enormously. The agonist and the antagonist work together or in a different way. And then if my arm is moving, then those muscles that in the future will work together and under the control of the brain, they send messages together in time. And this togetherness in time helps guiding the horizontal connections and making a map of your body. There is no, your body map is individualized, and your growth rate is different. So a baby has to learn to scratch the nose in a different way every day, because the distance between the finger and the nose is changing. So this has to be updated. So when the system is calibrated, so to speak, then your movement is no longer related to a spindle, because acetylcholine grows in and so on, but the system is already calibrated. Every night before you go to sleep, you will observe, not every night, every now and then you observe that you have this huge startle pattern, you almost fall off the bed. This is recapitulating your baby days or intrauterine life, that it generates a spindle during transition from one sleep stage to another sleep stage. So this is a beautiful way of saying what movement can give to the brain. And then just here we go further and further, and I use the example of the disengagement from movement from navigation to memory, which is the same kind of thing, saying that what calibrates the system is movement. Of course, in the adult, when the system is calibrated, you no longer need to act out. That's the beauty of it. You just have, thoughts about what if I do this, what if I do that, and every animal does the same way, with the two bigger differences. One is that a small brain is capable of predicting the future in a very simple environment and at a short time scale. And then we add a lot of complications, a lot of loops and things like that to the brain, all of which serves the same purpose, is predicting the future, but now in a noisy environment and over the time scale of days, years, and so on.
Yeah.
So this is, for me, a fundamental way of showing how you can go from action organization to cognition.
Yeah. Yeah, that's quite amazing stuff. So I think we covered a lot, Yuri, and appreciate you taking the time for this. I've learned a lot, and I hope our listeners and viewers do too. And I will, in the description section, I'll put some links to a few of your review articles and your lab and so forth.
Thank you. And I'm reading your books. I feel guilty every single time I eat something that I like.
So, but so all I do is skip breakfast. I used to actually, Kentucky, we lived 3 miles from the lab. So I'd get up early in the morning, like 5 o'clock, and I'd run physically. I'd run to the lab. So I get my exercise just running to the lab and back. Didn't eat breakfast, and I'm most productive in the mornings, always. You eat a meal, you eat a meal, you get sleepy.
My pathology professor told me that you can learn only when you can be effectively learning when you are hungry and cold. But
yeah, that's true.
But your suggestion is against the old wisdom that says that in the morning eat like a king, and for lunch, I don't know, it's like water, and for dinner you eat like a beggar.
But it wasn't based on science. Now we're going back to the.
So you recommend the opposite.
It's easy. Once you takes several weeks to a month. If you normally eat breakfast, if you stop eating breakfast, you're going to be hungry and irritable for a week or two. But then after that, gone. No more hungry in the morning.
Okay, well, try.
Okay.
Good
Thanks a
talking
lot.
to you. Take
Bye.
care. Bye.